Hello everyone, I am Toco Y P CHUI of the Neuroretinal Infirmary of Mount Sinai. First of all, I would like to thank the organizing committee for this wonderful opportunity to present to you today. I'll be sharing our study Optic Nerve Head Microarchitecture on-fast OCT and OCT angiographic imaging of lamina cribrosa, neuroretinal rim, and vasculature in glaucomatous eyes. I would like to thank our team members from the Neuroretinal Infirmary and our collaborators from the Medical College of Wisconsin and Columbia University. And these are our disclosures. Our funding sources are listed here. The purpose of this study was to image and quantify lamina cribrosa pores, neuroretinal rim area, and peripapillary capillary density changes in eyes with open-angle glaucoma using on-fast OCT reflectance and OCT angiography images. I have some sample images here from one of our glaucoma patients. As you can see, two distinct retinal nerve bundle defects on the on-fast OCT reflectance images on the left here. Again, we'll be looking at the on-fast images of the lamina cribrosa pores right here, neuroretinal rim, and capillary network around the optic nerve head using a clinical OCT device. A little background information. The lamina cribrosa is a porous connective tissue through which the retinal ganglion cell axons travel back to the brain. Lamina cribrosa pores are commonly visible in glaucoma subjects. They are also known to play a critical role in the pathogenesis of glaucoma. On the left is a SEM image of human lamina cribrosa. And for comparison, I have here on the right are two optic nerve head images from one of our glaucoma patients, imaged using fundus camera and a clinical OCT device. Glaucoma is also characterized by the progressive thinning of the neuroretinal rim. The loss of neuroretinal rim area has been studied using colorless fundus picture, cross-sectional OCT, and in the current study, we use on-fast clinical OCT images to quantify the neuroretinal rim area in controls and patients with glaucoma. So in this study, we imaged the lamina cribrosa, neuroretinal rim, and the peripapillary capillary network in 12 patients with moderate to severe glaucoma and 27 controls using a clinical OCT device. First, 10 sequential OCT reflectance and OCTA images were acquired using an after-view OCT. Images were then registered and averaged as described in our previous studies. From image acquisition to image processing, it takes about 3 hours in total. Here you can see a fly-through on-fast view of the registered average OCTA and OCT reflectance, and the corresponding axial locations are indicated by the horizontal lines on the cross-section OCT panels. Here we will be looking at an example from a glaucoma patient. This is a 4.5 millimeter cross-sectional OCT image across the disc in a patient with glaucoma. After image registrations and averaging, we extracted a 3 microns on-fast OCT reflectance slab located 30 microns below the inner limiting membrane surface, which is indicated on the left panel here. In this on-fast OCT reflectance slab, you can see this glaucoma patient had a very distinct retinal nerve fiber bundle defect at the inferior temporal region and a very mild retinal nerve fiber bundle defect at the superior temporal regions in here. Next, for the visualization of lamina cribrosa pores, we extracted another on-fast 3 microns OCT reflector slab located at 150 microns below the trough of the optic cup, which is shown on the right panel here. The corresponding axial location of this on-fast OCT reflected slab is indicated by the cyan straight line on the cross-sectional OCT image in the middle panel. On the lamina cribrosa image, we can now see very clearly the lamina cribrosa pores. We then cemented out the pores automatically after the removal of the larger blood vessels. The cemented laminar pores are highlighted in cyan color in here. Neuroretinal rim area segmentation was performed on a 3 microns on-fast OCT reflected slab located above the surface of the RPE, and the corresponding axial location of this OCT reflected slab is indicated by the yellow lines on the cross-sectional OCT. In this on-fast OCT reflected slab, you can see this patient had a very distinct neuroretinal rim thinning at the inferior temporal and superior temporal regions here, which is consistent with the retinal nerve fiber bundle defect as seen on the OCT reflected slab on the left panel here. In order to measure the neuroretinal rim area, we manually segmented the optic disc margin, which is indicated in cyan color on the right panel. We then manually segmented the neuroretinal rim inner margin, which is indicated in red. So the neuroretinal rim area was measured as the area between the red and the cyan margins, which is shaded in orange color on the right panel here. The last measurement was the peripapillary capillary density, which was measured on a 150 microns OCT angiography slab located below the inner limiting membrane surface. So this is a thicker slab which is shaded in green on the cross-sectional OCT image. After the removal of the optic disc center and the larger blood vessels, we measured the capillary density in percentage as indicated by the capillary density map on the right panel here. Here are the results. In control eyes, the mean of lamina cribrosa pore areas was about 2,700 microns square. The mean of neuroretinal rim area was about 1.65 millimeters square, and the mean of peripapillary capillary density was about 42% in controls. In glaucoma group, which is indicated in white in here, lamina cribrosa pore area was significantly larger as compared to the control group, whereas the neuroretinal rim area and the peripapillary capillary density were found reduced in eyes with glaucoma as compared to the control group. Here is a side-by-side comparison of the lamina cribrosa pores, neuroretinal rim area, and capillary density images from a control, which is on the top row here, and a glaucoma patient, which is on the bottom row here. Similar to the previous glaucoma patient, this patient also had a very distinct neuroretinal rim thinning at the inferior temporal and superior temporal regions here, which is consistent with the retinal nerve fiber bundle defect as seen on the superficial slab in here. So in general, the lamina cribrosa pore area is larger and the neural rim area is smaller in the glaucoma patients with reduced peripapillary capillary density. Using our image processing approach, we are also able to visualize the spatial relationship between the lamina cribrosa and its capillary network. And this is a 3D rendered lamina cribrosa structure, which is colored in green, and the corresponding capillary network, which is colored in red in a patient with glaucoma. This is the same region showing just the capillary network of the lamina cribrosa with the axial locations coded in different colors. Red color means more anterior and blue means more posterior. In conclusion, the on-fast OCT reflectance and OCT angiography are uniquely complementary for characterizing optic nerve head microstructure and microvasculature in glaucomatous and control eyes. The inverse relationship of lamina cribrosa pore areas with neurorim area and peripapillary capillary density in eyes with glaucoma is consistent with the current model of glaucomatous damage. Thank you for viewing this presentation. Everybody, please stay safe and well, and I'll see you in person next time. Bye.